in this video I am going to discuss about heteropolysaccharides. First, let's see an introduction to polysaccharides. Polysaccharides are high molecular weight carbohydrate polymers composed of many monosaccharide residues linked by glycosidic bonds. They may be linear or branched and undergo hydrolysis to yield monosaccharides or oligosaccharides. Characteristics of polysaccharides include they contain hundreds to thousands of monosaccharide units, and most of the polysaccharides are insoluble or sparingly soluble in water. They generally non-sweet in taste, and they are non-reducing carbohydrates because most anomeric carbons are involved in glycosidic bonds. They function mainly as storage carbohydrates or structural materials. Now coming to the classification of polysaccharides. Polysaccharides are classified based on the type of monosaccharide units present. They are classified into homopolysaccharides and heteropolysaccharides. Homopolysaccharides are polysaccharides that contain only one type of monosaccharide repeated throughout the molecule. Examples are starch, glycogen, dextrin, inulin, cellulose, chitin, etc. And heteropolysaccharides are complex carbohydrates made up of two or more different types of monosaccharide units joined together by glycosidic bonds. Unlike homopolysaccharides, which contain only one type of sugar, heteropolysaccharides contain different sugars arranged in repeating units. Heteropolysaccharides are mainly found in extracellular matrix of connective tissues, bacterial cell walls, plant cells, and mucous secretions. They perform structural, protective, lubricating, and regulatory functions. Characteristics of heteropolysaccharides include it is composed of two or more different monosaccharides and usually consists of repeating disaccharide units, and it has high molecular weight compounds, and most are branched or linear polymers. And these heteropolysaccharides are highly hydrophilic due to numerous hydroxyl and carboxyl groups. It forms a highly viscous solutions on shells, and it may contain uronic acids and amino sugars. It is often associated with proteins to form proteoglycans. Again, heteropolysaccharides are broadly classified into two types: glycous aminoglycans or mucopolysaccharides and non-glycous aminoglycans. First, we'll see about glycous aminoglycans. Glycous aminoglycans are long unbranched heteropolysaccharides composed of repeating disaccharide units containing one amino sugar and one uronic acid. And most of the glycous aminoglycans are sulfated except hyaluronic acid. There are six major types of glycous aminoglycans or hyaluronic acid: chondroitin sulfate, dermatan sulfate, keratan sulfate, heparan sulfate, and heparin. Now we'll see about hyaluronic acid. Hyaluronic acid is made up of repeating units of D-glucuronic acid and N-acetyl D-glucosamine. Unlike other glycous aminoglycans, it is not sulfated, making it unique. And these are found in the synovial fluid of joints, umbilical cord, vitreous humor of the eye, skin, and connective tissues. It is widely distributed throughout the body. And coming to the functions, hyaluronic acid is present in the synovial fluid of joints. They reduce friction between the bones and allow smooth and painless movement. It maintains tissue hydration by holding large amount of water. It also acts as a shock absorber in joints, helps in cell migration during tissue repair, and promotes wound healing, and also it supports embryonic development. Clinical importance of hyaluronic acid is used in the treatment of osteoarthritis to reduce joint pain. It is also used as a cosmetic dermal filler to reduce wrinkles and improve skin appearance. It promotes faster healing of wounds and damaged tissues. And the next one is chondroitin sulfate. It is composed of D-glucuronic acid and N-acetyl galactosamine sulfate. It is found in cartilage, tendons, ligaments, bones, and heart valves. These tissues require strength and flexibility. In functions of chondroitin sulfate, it provides tensile strength to connective tissues. It maintains the elasticity of cartilage and helps it resist compression. It also supports the normal function of joints. In clinical importance, it is used as a dietary supplement for osteoarthritis. During cartilage degeneration, its concentration decreases and leading to joint problems. Then third one is dermatan sulfate. It is composed of L-iduronate and N-acetyl D-galactosamine 4-sulfate. It is another sulfated glycosaminoglycan. It is found in the skin, blood vessels, arteries, and tendons. It is especially abundant in tissues that require flexibility. In the functions of dermatan sulfate, it helps maintain the elasticity of the skin. It promotes wound healing and tissue repair after injury. It also plays a role in regulating blood coagulation. The next one is keratin sulfate. Keratin sulfate is made up of D-galactose and N-acetyl D-glucosamine 6-sulfate. It is the only glycosaminoglycan that does not contain uronic acid, and it is present in the cornea of eye, cartilage, and invertebral discs. These tissues require transparency or flexibility. And coming to the functions, keratin sulfate is present in the cornea of the eye. It maintains the proper arrangement of collagen fibers and keeps the cornea transparent, which is necessary for clear vision. It also provides flexibility to cartilage and offers mechanical support to connective tissues. In clinical importance, a deficiency of keratin sulfate may contribute to corneal disorders and affect vision. It may also reduce the strength of cartilage. Next one is heparin. Heparin is composed of repeating disaccharide units of D-glucosamine and either of 2 uronic acids. And it is the most highly sulfated glycosaminoglycan. And it is mainly found in the mast cells, especially in the liver and lungs. It is released when needed to prevent clot formation. Functions of heparin: it acts as a natural anticoagulant in the body. They prevent excessive blood clotting by increasing the activity of antithrombin 3, thereby ensuring normal blood circulation. And clinical importance, it is widely used as an injectable anticoagulant in hospitals. It is used to treat deep vein thrombosis, pulmonary embolism, during cardiac surgery, and during hemodialysis to prevent blood clotting. Next one is heparin sulfate. Heparin sulfate has a structure similar to heparin but contains fewer sulfate groups. Therefore, it has less anticoagulant activity than heparin. And it is present in the basement membrane and on the surface of cells and in blood vessels. It is an important component of many tissues. Functions of heparin sulfate, which help cells attach to each other and to the extracellular matrix. It participates in cell signaling and regulates the activity of growth factors. It also plays an important role in the filtration in kidneys and support normal tissue growth and development. Another important one is proteoglycans. Proteoglycans are large molecules made up of a core protein with one or more glycosaminoglycan chains attached to it. The major components of the cell extracellular matrix and provide support to many tissues. Examples include aggrecan, decorin, versican, and syndican. And in proteoglycans, the protein provides the framework while the glycosaminoglycan chains perform most of the biological functions like shock absorption, cell adhesion, tissue hydration, structural support, and growth factors binding. Another classification of heteropolysaccharides is non-glycosaminoglycan heteropolysaccharides. In these heteropolysaccharides that do not belong to the glycosaminoglycan family, they are mainly found in plants, algae, bacteria, etc. And it has an important industrial and biological applications. The first one is pectin. Pectin is composed of galactonic acid along with rhamnose, arabinose, and galactose. These sugars form a complex polysaccharide present in the plants. And the sources of pectin is naturally present in cell walls of fruit and other plant tissues. It helps maintain the structure of plant cells. Coming to the functions, pectin strengthens the plant cell wall and helps maintain its structure. and it retains water in plant tissues and prevents excessive water loss. And during fruit ripening, pectin is broken down by enzymes, making the fruit soft and edible. And the uses of pectins are pectins act as a natural gelling agent and helps jams and jellies set properly. In pharmaceutical, it is used as a pharmaceutical product to prepare gels, tablets, and control drug formulations. And the next one is agar. It is composed mainly of galactose and sulfated galactose. These sugars give agar its strong gel-forming property. And the sources of agar is obtained from red algae. It is widely used in laboratories and the food industry. And coming to the uses, agar is used to prepare solid culture media for growing bacteria and fungi because most microorganisms cannot digest it. And in food industries, agar is added to food as a thickening and stabilizing agent. In pharmaceutical preparations, agar is used in pharmaceutical formulation as a suspending, thickening, and stabilizing agent. And the next one is alginic acid or alginate. Alginic acid is composed of mannuronic and glucuronic acid. These sugars give alginate its gel-forming ability. Sources of alginate is obtained from brown algae such as kelp and seaweed. And coming to the uses, alginate is widely used by dentists to make accurate impressions of teeth. And alginate is added to foods to improve texture and stability. And alginate is used to prepare controlled drug delivery system because it forms biodegradable gels. In wound dressing, alginate dressing absorbs wound fluid and keeps the wound moist, helping it heal faster. Next we'll see about peptidoglycan. Peptidoglycan is made up of alternating units of N-acetylglucosamine and N-acetylmuronic acid, and it is linked together by short peptide chains. This forms a strong mesh-like structure. Peptidoglycan is present in the cell wall of bacteria and it is absent in animal cells. And coming to the functions, it provides peptidoglycans give strength and rigidity to the bacterial cell wall. And it also protects bacteria from bursting due to osmotic pressure when water enters the cell. And it also helps bacteria maintain their characteristic shape such as spherical or rod-shaped forms. And clinical importance of peptidoglycan is it acts as many antibiotics act by inhibiting peptidoglycan synthesis and weakening the bacterial cell wall. And the antibiotics include penicillin, cephalosporin, vancomycin, etc. And the final one is clinical importance of glycosaminoglycans. And the first one is osteoarthritis. It is a degenerative joint disease in which the cartilage gradually wears away. During this condition, the amount of chondroitin sulfate in cartilage decreases, making the cartilage weak and less elastic. The concentration of hyaluronic acid in the synovial fluid also decreases and reducing joint lubrication. As a result, the patient experiences joint pain, stiffness, swelling, and difficulty in movements. The next one is mucopolysaccharides, or a group of inherited lysosomal storage disorders. They occur due to the deficiency of the enzyme required to break down glycosaminoglycans. As a result, glycosaminoglycans accumulate inside the lysosome of cells and damage various organs and tissues. Examples include Hurler syndrome and Hunter syndrome. Hurler syndrome is caused by the deficiency of the enzyme alpha-L-iduronidase. Because of this deficiency, dimethyl sulfate and heparan sulfate accumulate in the body. In Hunter syndrome, it is caused by the deficiency of the enzyme iduronate sulfatase. It also leads to the accumulation of dimethyl sulfate and heparan sulfate. But unlike Hurler syndrome, corn meal clouding is absent. And the next one is anticoagulant therapy. Heparin is naturally occurring glycosaminoglycan that acts as a powerful anticoagulant. It prevents the formation of blood clots by activating antithrombin 3, which inhibits clotting factors. And the next one is cosmetic and ophthalmic uses. Hyaluronic acid is widely used in medicines because of its excellent water retaining and lubricating properties. In dermal fillers, it is injected into the skin as a dermal filler to reduce wrinkles, restore facial volume, and improve skin appearance. In eye surgery, it is used during eye surgery such as cataract surgery to protect delicate eye tissues and maintain the shape of the eye. And hyaluronic acid is added to artificial tear solution to lubricate the eyes and release dryness and irritation. And in osteoarthritis injections, hyaluronic acid is injected into the affected joints to improve lubrication, reduce pain, and increase joint mobility in patients with osteoarthritis. Thank you viewers for listening and watching my video.